found out about the DSP. In fact, I've been looking for a while for a technology that can give us the answers that we want. And there really isn't anything out there. So we've been looking initially at sequencing technology, single-cell bulk sequencing technology. And while that gives you a lot of data, it doesn't tell you anything about the cells in terms of where they are. And that type of architectural detail and spatial detail is critical when you're looking at tissue samples, and it's particularly looking at biopsies of kidneys and so on. On the other hand, looking at the traditional histological immunohistochemical techniques, it just doesn't give you the depth of data that you need. So the focus of a grant of our NanoString DSP grant application was to look at patient biopsies from patients who are receiving a very novel cellular therapy, which is regulatory T-cell therapy in renal transplantation. And it's one of the largest clinical trials of its kind in the world. And it's a randomized clinical study where a group of patients will either receive regulatory T-cell therapy or not. And the ones that receive the regulatory T-cell therapy will have their immunosuppression withdrawn. So it's looking at the biopsies from these patients and determining whether there's a signature of immune tolerance. So hearing that I won the grant was fantastic news. I mean, that made my day, week, month, made my year. Absolutely fantastic news, but actually what it means is that now we can analyze the samples in our study to a degree of clarity that we couldn't before, and that is absolutely key for these clinical studies where tissue samples are invaluable and rare and need to be looked after and can't be used from more than one analysis technique. I think that's key. And being able to get as much data as we can out of these tiny pieces of tissue that really directs the next stage of our clinical studies. So I think that is absolutely fantastic, being able to look in such detail, see if there's a signature of immune regulation or tolerance in these tissues, and to be able to differentiate how different patients do in response to therapy. There's nothing else like it. It will open doors to new collaborations for certain, and it will cement collaborations that we already have. And it will clearly make us much more competitive and give us the edge in terms of new grant applications. But really, this is all for the benefit of science and for patients, and I can see how adding all this information will really give us a much more clear idea of how patients are doing in response to these very novel therapies, and that is very important.